Hi, my name's Tori Gill. I'll be walking you through my DNA extraction of split peas. So DNA is deoxyribonucleic acid. So basically you have your nitrogenous base, a ribose sugar, which is a pentose sugar, um, and a phosphate. Deoxy meaning the 2' carbon is deoxygenated. Um, that's DNA, and then RNA will have both the uh, 2 and the 3 will remain oxygenated. Um, so DNA is the genetic material for all living things. Each triplet codon of bases codes for a specific amino acid. So we'll go ahead and start with the first step of the procedure, which will be physically breaking down the split pea tissue. You can use a blender or mortar and pestle. I'm going to opt for the blender method. So we'll take our split peas, and that's 60 milligrams of split peas. We'll put both salt packets in. This is just regular table salt, sodium chloride. The reason for the salt is so, as I just said, DNA has that backbone of sugar phosphate. Now the phosphates are negatively charged, so in water, which we'll put a cup of water in next, they'll repel each other. So you'll just have like single strands of DNA, be hard to identify. Now, if you add sodium chloride, the positively charged sodium ions will kind of clump the DNA together. Instead of repelling each other, they'll clump together. So you'll get a better precipitate that you can identify. All right, and then we'll add 1 cup distilled water. All right, I'll go put these through the blender. I'll be right back. Split peas in a sodium chloride solution, basically. We'll filter them, filter the DNA extract through this cheesecloth. Okay, you can see we have some solid up here, and the extract went through the cheesecloth. Yeah, so since this layer on the bottom is our pDNA extract, we can get rid of this cheesecloth and the solids. So now what we'll do is add dish soap to the DNA extraction. And with this detergent, we'll do, so we still have to further break down, we physically broke down the peas, but now kind of biochemically, we have to break down the cell membrane and the nuclear membrane um, for cell lysis so that we can get to that DNA. So, as you know, the cell membrane is made up of lipids and proteins. Detergent can, what's the word, penetrate that cell membrane because, like the bilayer, detergent is both hydrophilic and hydrophobic portions to it. and it can also denature the proteins in the cell membrane. So we'll use this whole dropper. Now we'll put this solution in the fridge for 10 minutes. When it's in the fridge for our DNA extract is done, taking it out. See if you can see it. There you go. So what we're going to do now, we're going to transfer it to our 150 ml test tube. So we're going to go about 1/3. So if we break this up into thirds, say about right there. 1, 2, 3. So say that's about good, right? 1/3. I'm using this glass to hold it. Okay, so we'll put our extra right here. And there's our test tube with 1/3. So now what we're going to do, we're going to add the papain powder. We're going to add half of it. There's not much in there, so we'll just do a couple dabs, get some flakes in there. So what this is going to do, this powder is a protease, so breaks down or catalyzes the breakdown of proteins. So what we're trying to do is break down those histones that the DNA is tightly wound around so that they can precipitate. Okay. That's about half. So we'll make sure to gently stir with our clean rod. We've cleaned it. We'll gently stir so not to break up any of the DNA. Alright, so now it's time to add our isopropyl alcohol. We'll go get that from the freezer. So here's 91% isopropyl alcohol that's been in the freezer overnight. Now we'll add our 91% isopropyl alcohol. It's going to be at the same ratio as the DNA extract, so basically we'll just add another third. So we should be 2/3 along the test tube. So I'm going to use this bottle syringe to kind of control the alcohol going in. So what we'll do, hold it at a 45-degree angle, slowly drop isopropyl alcohol on the side and let it drop down on the side of the test tube. And the reason why we're adding this chilled isopropyl alcohol, so I mean, think about the best solution to dissolve something in, right, is always hot water. Super soluble, cold water, not as well. Cold alcohol, the DNA will be completely insoluble in that solution. So it will have precipitate. It won't dissolve in the solution. Okay, and there's our about 2/3, 1/3, 2/3. You can see the separation. So I don't know if you can see it as well in this camera, but maybe in this one it's a little clearer. So I don't know if you can see it, but you can see small white precipitates floating, and you see these strings coming off of the DNA extract. So DNA is separating, it's starting to precipitate up at the top, and that's how we're able to separate DNA from the actual P extract. But we'll let it sit for about a minute to see if we can get some more precipitation. It's not quite what I expected to see. I expected to see more DNA than this small layer we have here. Um, but I think that might have been due to my blending technique. It was uh, quite a large blender for such small split pea samples. I think next time I'd prefer to use the mortar and pestle method to have a little bit more control over the physical tissue breakdown. But we do have a good layer here. You can see it better here. We have a pretty good layer, and you can see the DNA separating really good on this side. You see quite a bit of separation. It's starting to precipitate out. So the DNA looks white. It has bubbles accumulating, and it seems to be separating from the solid organic layer right here into the isopropyl alcohol. And it seems somewhat stringy. It is clumpy, but it's like clumps of strings, which is what I expected to see as far as the actual shape. But it's white, and oh, I see one small precipitate here climbing up, but it's starting to separate a little bit more, which is what I wanted to see.